When we hear of the word bank, we commonly think of different types of financial banking institutions. However, money is not the only resource that is of vital importance to humans. In fact, there is a bank only for the storage of seeds. The picture shown is the Svalbard Global Seed Bank, which is also known as the Doomsday Vault. It is a seed vault that safeguards the world's most diverse collection of seeds. The vault will secure millions of seeds representing every important crop variety available in the world today. This acts as the final backup when future generations face the challenges of climate change and population growth. In this video, our goal is to name the stages of embryo development as well as endosperm development. We also explain the importance of dormancy and compare seed germination in monocots and dicots. So the seed develops from the fertilized ovule and includes an embryonic plant and some form of nutritive tissue called the endosperm enclosed in what we call as the seed coat. Therefore, seeds contain the next generation and so completes the life cycle of a flowering plant. When mature, a seed consists of a dormant embryo surrounded by stored food and protective layers. After successful pollination and double fertilization, a seed begins to form. Recalling double fertilization, one sperm fertilizes the egg cell forming a diploid zygote, which will later become the embryo, and the other sperm fertilizes the central nuclei, which will become the triploid endosperm. In the next slide, we will discuss how the endosperm, which is the tissue that surrounds and nourishes the growing embryo, forms or develops. The endosperm usually develops first before the embryo. The central nuclei divide to form a multinucleate supercell with a milky consistency. It is important to note that the type of division happening in the endosperm is nuclear division, not cellular division. Why? Because there are no cell walls that encloses these nuclei just yet, which also explains the liquid milky consistency of the endosperm. As the endosperm increase in size through nuclear division, dense cytoplasm gathers around the nuclei and walls are constructed, thus forming cells. Take the coconut as an example. The coconut milk and meat are examples of liquid and solid endosperm respectively. In the early development of a young coconut fruit, the endosperm nuclei divide and increase in number. At this time, the endosperm of the coconut is in liquid consistency because the nuclei that makes up the endosperm do not still have partitions brought by the cell walls. As the coconut ripens, the liquid endosperm begins to form walls around individual nucleus, causing it to harden and become solid. While in grains and most other species of monocots, the endosperm stores nutrients that can be used by the seedling after germination. So the endosperm of grains such as corn, wheat, and rice occupies the bulk of the kernel and is the main energy reserve for the development of the young seedling. A monocot stores the bulk of its energy in the endosperm. In contrast to dicot seeds, the food reserves of the endosperm are completely exported to the growing embryo as the seed completes its development. For this reason, dicot seeds lack endosperm upon maturity. Instead, what provides nourishment to the embryo is the stored food in the cotyledons, not endosperm. So we now examine the different parts that make up the seeds. The embryo is the young multicellular organism before it emerges from the seed. The endosperm is the source of stored food consisting primarily of starches. The seed coat consists of one or more protective layers that encase the seed. The mature embryo also consists of an embryonic root known as the radicle, the embryonic shoot known as the epicotyle, and the junction between the roots and shoots known as the hypocotyle. Lastly, the cotyledon is described as a seed leaf that stores food in the form of starch and protein for use by the embryo. An embryo of a monocot plant has one while that of a dicot plant has two cotyledons. Following the development of endosperm, we discuss the development of the embryo. The fertilized egg, the zygote, divides mitotically and splits to form the basal cell and the terminal cell. In the basal cell, this gives rise to the suspensor, which anchors the embryo to the parent plant. The suspensor helps in transferring nutrients to the embryo from the parent plant and in some species from the endosperm. As the suspensor elongates, it pushes the embryo deeper into the nutritive and protective tissues. The other type of cell is the terminal cell, which provides or gives rise to the proembryo. Proembryo means early embryo. In here, cotyledons begin to form as bumps. A eudicot embryo with its two cotyledons is heart-shaped at this stage. As the embryo elongates, shoot and root apex appears. Shoot apex appears between cotyledons and root apex appears just before the tip of suspensor. The apices of shoots and roots sustain primary growth. Now we discuss seed dormancy. At some point in seed development, hormonal signals tell cells in the embryo and endosperm to stop dividing, and the seed gradually loses moisture and enters what we call dormancy. Seed dormancy is a survival mechanism by which seeds can delay germination until the right environmental conditions for seedling growth and development. This is a crucial adaptation because it ensures that seeds have time to disperse away from the parent plant before germinating. Moreover, dormancy enables seeds to postpone development if the environment is unfavorable, such as during drought or frost. So, favorable conditions trigger embryo growth to resume when young plants are more likely to survive. The requirement for specific cues to break seed dormancy increases the chance that germination will occur at a time and place most advantageous to the seedling. Seeds of many desert plants, for instance, germinate only after a substantial rainfall. If they were to germinate after a mild drizzle, the soil might soon become too dry to support the seedlings. Where natural fires are common, many seeds require intense heat or smoke to break dormancy. Seedlings are therefore most abundant after fire has cleared away competing vegetation. Where winters are harsh, on the other hand, seeds may require extended exposure to cold before they germinate on the next season. Lastly, certain small seeds such as those of some lettuce varieties require light for germination and will break dormancy only if buried shallow enough for the seedlings to poke through the soil surface. Most seeds are durable enough to last a year or two until conditions are favorable for germinating. Thus, the soil has a bank of ungerminated seeds that may have accumulated for several years. This is one reason vegetation reappears so rapidly after an environmental disruption such as fire. So, germination is the resumption of growth and development after a period of seed dormancy. With appropriate environmental conditions such as adequate moisture, oxygen, and appropriate temperature, seeds can germinate. Seed germination is initiated by imbibition, which is the uptake of water due to the low water potential of the dry seed. Imbibition causes the seed to expand and rupture its coat and triggers changes in the embryo that enable it to resume growth. Following hydration, enzymes digest the storage materials of the endosperm or cotyledons, and the nutrients are transferred to the growing regions of the embryo. As the seedling soon begins to take on its mature form, young roots grow downward in response to gravity, anchoring the plant in the soil and absorbing water and minerals. The shoot produces leaves as it grows upward toward the light. Initially, the energy source for the seedling's growth is stored food inside the seed. By the time the seedling has depleted these reserves, the new green leaves should begin producing food by photosynthesis. But if a seed is buried too deep in the soil, the young plant will never emerge. In effect, it will starve to death before reaching the light. We now compare the germination between the dicot and monocot seeds. In dicots, the first organ to emerge from the germinating seed is the radicle, which is, as you can recall, the embryonic root. The development of a root system anchors the seedling in the soil and supplies it with water necessary for cell expansion. In garden beans, for example, a hook forms in the hypocotyl, and growth pushes the hook above ground. In response to light, the hypocotyl straightens, and the cotyledons separate, and the delicate epicotyl, now exposed, spreads its first true leaves. These leaves further expand, become green, and begin making food by photosynthesis. The cotyledons shrivel and fall away because their food reserves have been exhausted by the germinating embryo. In monocots, such as corn and other grasses, they use a different method for breaking the ground when they germinate. Instead of a hook like that of in dicot, the coleoptile, which is a sheath protecting a young shoot tip, pushes up through the soil and into the air. The shoot tip grows through the tunnel provided by the coleoptile and breaks through the coleoptile's tip upon emergence. The sheath are discarded when the seedling begins growing, and the immature leaves become the first true leaves of the corn plant. So I'll leave you with this question. Does seed size matter? The size of a plant's seed reflects an evolutionary trade-off. Large, heavy seeds contain ample nutrient reserves to fuel seedling growth but may not travel far. On the other hand, small seeds store limited nutrients but tend to disperse far and wide. So to answer the question, does seed size matter? The answer is yes, but neither is superior nor inferior over the other. It all depends on what environmental conditions call for a large seed or a small seed as far as plant growth is concerned. So we summarize or we wrap up everything that was learned in this video. So first, seeds develop from ovules. A seed consists of an embryo, an endosperm, and a seed coat. In double fertilization, one sperm cell nucleus fertilizes the egg to form the diploid zygote, and the other fertilizes the central cell's two nuclei, forming the triploid endosperm. The zygote undergoes a series of developmental stages to become an embryo. In dicots, the embryo has two cotyledons, which absorb the endosperm, and in monocots, the embryo has a single cotyledon and endosperm. Further, seed dormancy ensures survival of seeds. Seeds germinate only when conditions for seedling survival are optimal. The breaking of dormancy often requires environmental cues such as temperature or lighting changes. Next, plant growth begins with seed germination. Given water, oxygen, and a favorable temperature, a seed germinates. The embryo bursts from the seed coat and its development resumes. Lastly, seed germination varies in monocots and in dicots. In a monocot such as a corn, a sheath-like coleoptile covers the shoot until the first foliage leaves develop. The cotyledon remains below the ground. On the other hand, in green beans and some other dicots, the cotyledons emerge from the ground. They carry out photosynthesis until the first foliage leaves form.